We now have our second keynote talk for TransMed, and it gives me great pleasure to welcome Professor Tim Hubbard from King's College London, who will be giving the second keynote. Tim is head of the Department of Medical and Molecular Genetics at King's, and he is also Director of Bioinformatics for King's Health Partners. Tim also has a secondment, a part-time secondment, as head of the genome analysis at Genomics England. Genomics England is the company set up by the Department of Health to deliver the 100,000 Genomes Project to sequence and analyze the genomes of 100,000 patients, focusing on cancer and on rare diseases. So Tim's PhD and his postdoctoral work, his early work, was in protein structural bioinformatics. And he co-founded the SCOP database, the structural classification of proteins database, which is a very well-used resource by structural bioinformaticians. Tim was also involved for many years in the organization of the Protein Structure Prediction Challenge, the CASP competitions. And previous to his position at King's College, Tim worked at the Wellcome Trust Sanger Institute for 16 years, where he was head of informatics. And Tim is going to talk to us about the 100,000 Genomes Project and personalized medicine, and it gives me great pleasure to introduce Tim. Thank you very much.
Thanks very much. It's very nice to be here. So this is probably a very different talk to the others in this. I'm not really going to talk about any scientific results. I'm going to talk about how you bring together the health system and research. Up to now, the way people have done things is cohorts of patients who are research subjects. But of course, that's always limited by budgets available for research subjects. But then you've got the whole health system. Can you instead try and bring this whole thing together so every patient becomes, their data is available for research, but similarly, every bit of research you do potentially can feed back and help the patient in their own personalized care. So this is the kind of learning health system, and this project is kind of acting as a starting point for building that sort of thing. Now, obviously, history of this, there's human genome was done quite a long time ago now and costs a huge amount of money, and I don't think anybody thought that we would get down to these prices of $1,000 per genome, but that's really what's driven the idea that you can start doing whole genome sequencing in a health system. And that led in the UK to lots of discussions, things like the House of Lords produced a report saying we should look out for genomics getting to a point where it can be deployed in the health system. And in 2012, after the Olympics, as a legacy of the Olympics, the Prime Minister announced this 100,000 Genomes Project. 6 months later, Genomics England was formed to execute this. What was it going to do? So, of course, many people are thinking, I get a whole genome, I can do prediction. This is very much not what it's doing. It's specifically targeting low-hanging fruit where you can actually take a genome and say something useful for that patient directly. So take patients who are ill with particular diseases, rare diseases and cancers, and analyze the genome from the point of view of diagnosing their condition. So the project as a whole is a treatment project. It's actually officially got a designation as an Nhs Transformation Project. NHS is the National Health Service of the UK. So we're trying to change the health service to make it possible to do genomics just as another diagnostic test, to make it perfectly natural in the system for that to be a way of doing business. So I said, everything is whole genome sequence at 30x, so clinical grade sequencing. For rare diseases, we're not just sequencing the affected individual, we're sequencing the family, if we can get those family members as well. Cancer, we're sequencing the tumor and the normal. The timeline of this, I think the only useful thing to say is pilots were done initially using these kind of infrastructures we have where you bring universities close to hospitals, our so-called biomedical research centers. But after doing the pilots, we were told very firmly by Chief Medical Officer that in future everything has to be integrated directly with the health system rather than being seen as a sort of special research project. So that's what we've been doing in terms of setting this infrastructure up. So this project's got a number of objectives broader than just doing some science or treatment. We're trying to improve the health of these patients, this large number of patients. We're trying to stimulate wealth in the economy by stimulating genomics SMEs, things like that, small, medium-sized enterprises in terms of interpreting genomes. We're trying to create a legacy of infrastructure to support the whole health system. And then we're trying to enable research as well, make sure this data is also structured, the infrastructure is structured in a way that you can also do research using that data. So this is what we've built. So you have Little Genomics England. It's really like a contracting organization. It's not trying to build everything from scratch. There are lots of project managers and lawyers. And that's really how the project's being constructed. So the first thing was to get some sequencing capacity. We're not buying sequencers. We contracted with Illumina. Illumina in this contract have to do all the sequencing in England. So they had to expand their facility to be able to do this. And the contract is based on quality. So we have a quality metric. We have a certain bioinformatics capacity inside Genomics England for assessing all the data that comes back. And it's up to Illumina to meet these quality standards and the fixed pricing point as well. Now you have to have an interface with patients. For that, we're using the entire health system. So Nhs England is the organisation which runs the whole health system in the UK. 110 billion pounds a year flows through that organisation. They arrange contracts with hospitals to create genome medicine centres. So we now have a nationally deployed set of genome medicine centres, which actually involves around 85 hospitals. It's about a third of the health system that are now places which are certified for sample collection to certain standards and required to collect the associated clinical data. So you have the structure like this, where they also handle, of course, the consents of these patients. Then you've got to store this data somewhere. This is one of the key points, is what do you do in terms of data to make sure that you can use that data for clinical purposes and also use it for research. So firstly, you could build a data center, but nobody builds data centers anymore. Many people would nowadays say, well, you put this in the cloud. But of course, putting things in the cloud is quite controversial in terms of data security and privacy and how the public and patients feel about that. So actually, we're using a government framework. The UK government has a secure data center, but it has a contracting framework where you can get private cloud providers to install equipment inside this secure data center. So you get the advantages of cloud pricing, but within a building which is actually on a military base, which the UK government controls. So we can say this is inside the NHS firewall. It's inside the health system, this data center, and nothing moves outside that data center. All data must remain there. So here's the flow. DNA goes to Illumina, and BAMs and VCFs, because that's part of the contract to do the initial alignments and calling, goes to the data center. Another factor, of course, is this phenotype information. So everybody who's dealt with the medical system knows that getting the phenotype information out of the EHRs, the patient records, is a major challenge. But here we have a contract with these genome medicine centers. That's part of the contract. They have to either use an interface, open clinical interface, to enter structured data. There's a data model for every condition that's part of the 100,000 Genomes Project, or they have to learn to write XML to this particular standard. So, and you laugh, but actually the health service IT is learning to write XML. And because it's very painful to re-enter all the data in an open clinical web interface. And so this is putting lots of effort. There's lots of other pressures from Nhs England to move the whole of the NHS to SNOMED CT. We're requiring some of the terminology to be collected in HPO, but one of the particular things is there is a data model, there is a model data catalog which has all the diseases that are eligible with an exact data model that is part of the requirement of this contract to collect. So that's how we're getting the phenotype data. So once you've got that in there, you want to, well, the first phase is you want to interpret these genomes for these conditions. Again, you can build a pipeline. Many people here will have built these kind of pipelines to interpret variants. But we don't want to own a pipeline because, you know, who knows if we're going to be able to keep it to be the best one. We'd prefer to run an evaluation of different people, different academics or companies who are prepared to offer that service. So we ran a competition of 30 different groups took part. We've ended up contracting 3 companies who are providing clinical services. And again, the data doesn't go to them. They have to make their software run inside our data center. And that produces a clinical report, which goes back to the NHS, to the clinicians. Now, there's a little thing I missed there, grants here. So actually, part of this industrial strategy of stimulating wealth in the UK involves us running some inward investment into a range of SMEs, small medium enterprises in the UK, to stimulate the interpretation industry. And that's led to a few startups, you know, being transitioned to significant players. One in Oxford, one spin out of Sanger, and a few others as well. So that's all part of the overall strategy. Now, so that's where we are. This is the cycle now just for doing clinical interpretation. Where have we got to? As of, this is a bit out of date now, 22,000 whole genome sequences from patients and families. We've returned more than 2,000 reports, family-based reports, to the health system, about 1,500 in rare diseases and 500 in cancer. Now, why the discrepancy? Well, of course, this is the sort of flow chart of, you know, the kind of LIMS type flow chart of where everything is. We actually collected more samples than that than we've actually sequenced, but there's all kinds of stages along the way, and things, particularly getting all that clinical data and getting discrepancies resolved. You know, this is still work in progress in terms of construction. And certainly the interpretation side needs to speed up quite significantly. It's not a continuous flow pipeline yet. Now, what are we actually returning? So I think the key message is we are not taking a whole genome and making a whole range of predictions about a person's future. We're very narrowly interpreting for the condition which they're ill. They are in the health system. They've been through the existing diagnostics in the health system. They have unmet clinical need because they didn't get a diagnosis. They then become eligible for this program if it's in the list of diseases and has a clear phenotype. We're not sequencing people who the doctor thinks has something possibly genetic. It has to be much more defined than that, because then there's a chance of us producing an interpretation. Now, what about secondary findings? Of course, you scan a whole genome. There could be other significant things in there. That's optional in the consent. So patients are all consented. They can choose if they want to have additional secondary findings or not. And that list is very, very short. It's only things with very high penetrance and where there's something that can actually be done to help that patient in the health system. So it's much shorter than the American list, for example. There's also carrier status, which is fed back to parents, if that's applicable. All of this is governed through public patient engagement. There's a panel, and the representatives sit on all the governance committees of Genomics England. So there's a very serious engagement around how the consents are constructed, the documentation, the engagement with both patient groups and public in general about this sort of program, to make sure people are comfortable with the way it's being run. But one of the key points is this idea that the data, these are not really proper research subjects in the same sort of way as you set up a cohort. The deal here is you're sharing your data for additional analysis, but it doesn't move out of the health system. Now, in terms of recruitment, it's heavily biased at the moment in rare diseases to intellectual disability. But we've now got a substantial number of disease areas where we've got a significant number of genomes potentially available for analysis. Now, the informatics, so I've mentioned the clinical data models. I've mentioned these companies that are providing the interpretation. Now, if you've got 3 companies providing interpretation with 3 different interfaces, of course, that's a problem for the health system in terms of training of staff, etc. So we do try and wrap that in some sort of standardized framework, and that's around labeling the variants that have been chosen according to whether those variants fall into panels of genes which are known to be associated with that condition. So we have a crowdsourcing mechanism for gene panels, which is freely available. Anybody can log in, can use that system. So we end up before, to be eligible, the group that wants that disease added to the genomics list of diseases, they have to produce a gene panel that goes with that. So variants get labeled tier 1, 2, and 3. Tier 1 means it's a pathogenic variant in a database somewhere. Tier 2 means it's not labeled as pathogenic, but it seems to be significantly altering, and it's in one of these genes that's identified as being possibly associated with that condition. And then tier 3 is everything else. So this is just to guide clinicians when they look at these reports that come back, a standardized way of grouping these variants, despite the fact that there's 3 different companies providing the output. And all of this, there's an integrated system that is being built, partly by Genomics England, to just help clinicians handle all these variants and deciding what the result is and what they're going to do in terms of future clinical care. So these third-party providers feeding into a knowledge base, but integrating with, having an integrated framework. So there is actually a reporting panel which allows them to see the family pedigree, look at the reports, and do this sort of reviewing and decision-making on the actual variants. Now, what are we getting out of this? So obviously, you do a certain number of reports, deciding how many of them are accepted by the clinical community as being probably causal is a slower process. It has to go through multidisciplinary teams inside the genome medicine centers, inside these hospitals. But the numbers we have so far suggest that we're getting another 20% immediately, and a certain fraction on top of that. Of course, these are patients where they're the ones that didn't get diagnosed in the health system using the standardized processes. So this is an additional 20% on top of the 20%, say, that the health service already gets from single gene or panel tests. So it's bringing us up to, a significant fraction of patients being diagnosed. Now, I'll give you a couple of examples. Here's a case where a family, developmental delay case. It's a typical case in the sense that the patient has multiple conditions, has ended up seeing multiple specialist services with no conclusion, and then a whole genome sequencing finds a novel gene which was not recognized as a disease gene before, and that ends the diagnostic odyssey for that patient. And that's a major thing for the individual patients, because no more chasing around hospitals trying to work out what's wrong with that individual. Sometimes it actually produces something useful that you can act on. So here's another case, 4-year-old Jessica, who had developmental delay and seizures, and has similarly gone through a series of tests. We found a truncation of it's actually a glucose transporter gene into the brain, and that actually suggests a therapy. It's a glucose deficiency. And so she's been switched to a ketogenic diet, and that's improved her condition significantly. So this isn't the sort of thing that you would do normally, but having a whole genome analysis, finding a novel, clear novel deletion or truncation can suggest an alternative treatment path. And here's a third one. This is a case of a child that died with an unexplained condition, and it became clear that it was a problem with vitamin B12. In the meantime, the mother became pregnant again, and the second child, it was possible to put them on a vitamin B12 supplementation immediately. They have the same deficiency, but the child is doing fine. The child will have to stay on this probably for their lifetime, but this is a reason actually for doing rare disease analysis quickly, because sometimes you can intervene, possibly also with Jessica. If you had switched the diet earlier, you would have got an improvement over her condition, which is probably set after 4 years. So these are the kind of examples. You don't necessarily get, treatment for many of these patients. Rare disease, drug development is, fairly early on, but there is a possibility of understanding exactly what's wrong with the patient in a different sort of way. Now, cancer is proving much more difficult, partly because of the way that cancer samples, tumor samples, have historically been collected in a health system. The standard way is to put the slice of tumor tissue in formalin and fix it so the pathologist can then look at that and do their diagnosis, but that's not very good in terms of doing whole genome sequencing afterwards. We've spent a long time trying to fix that, change the protocol for extraction from the FFPE, but it doesn't seem to make a good quality sequence. So the whole of the NHS is now in the process of switching the entire system to fresh frozen. That means changing pathology services, changing theatre processes across the health system. It's being done in some accelerator centres initially, but that's, this is transformation of a health service to do that scale of switch. But we have, in the meantime, collected a significant number of samples, several thousand now, and there is also a diagnostic pathway, a report generating pathway. For cancer, we actually couldn't find a company that was producing sensible reports, so we, for the moment at least, have had to build one. But it's similar to the rare disease in the sense that variants are labeled according to whether they're kind of known or unknown, and it's linked to various sort of web resources that identify variants involved in particular conditions. So this is a sort of, it's the kind of limitations of what you can do in cancer right now, linking to particular information on public websites and clinical trials reports, because some of the options for treatment are to put an individual on a particular clinical trial. But it is proving to be useful. So this gives an idea of the reports we've returned so far. It's quite a high percentage that the ones in red are the ones where we have a kind of actionable gene output from that can be useful for the clinician in terms of deciding what to do. Now, I keep mentioning clinicians. Part of the transformation, of course, is training. You have to transform everybody inside the health system. Medical courses, like the course at King's, is being re-engineered to have a much stronger genetics component. But what about the 1.4 million people who are in the health system right now as part of staff at all levels? So we have something called Health Education England, which is another separate component of the NHS, and it's got a whole series of activities around improving genetic training. In fact, I noticed that NHGRI put it on their website as a YouTube video recently from this. So if you want to read more, you can go and find that YouTube video. But particularly, there's an MSc course, some of which has been standardized and is being taught now by 10 different universities across the country. MSc seems quite heavy, but this is structured as a set of modules, and the health service staff can take combinations of those modules in different ways as part of their professional training to become more, familiar with genome medicine. So that's the training angle that we're pushing. Now, research, which is probably more what you're interested in. So I said that everybody's consented. They're consented on this basis that the data gets used for collective analysis, partly to benefit them, because we're still maybe only getting, we're less than 50% diagnosis rate overall. But they also get consented for recontacting, so consent for contact. They get an additional sample which gets taken and put in a biobank, on the understanding that they're still part of clinical care. So anything that gets discovered on the research arm will get fed back to them if it's relevant for their treatment. So this is the model I had before. This is how it's extended. So now you have researchers. Researchers follow exactly the same rules. They can't take the data away. They have to work inside this data center, regardless of whether they're companies in the Gene Consortium or the researchers in the Genomics England Clinical Interpretation Partnership. There's this additional data. Patients have also consented for their entire long-term clinical records to be put in that data center as well. Everything in that data center is, of course, anonymized. And then there's this biobank sample, which researchers can access. We're not doing anything, Genomics England is not doing anything with that sample under our standard budget. but researchers, of course, can raise grants to allow something to be done to that sample. I would say that the researchers, again, don't get the sample. They will ask what they want to do with it, and Genomics England will contract something to happen and the data to appear in the data center, because these are all patients and the data is being controlled in this way. Already we've got, this is 22,000 people, we've got about 1.4 million episodes, hospital episodes, statistics incorporated. We have permission under this consent to have large amounts of data from across the health service integrated in this data center. So essentially, for these individuals, you're going to have the full medical life history available. Similarly, on the use of the biobank sample, there's some pilots going on in terms of what you can extract. The sample's already been split in a way that's compatible with RNA transcriptomics, for example. There's some options to do. Mouse Phenotype Consortium, for example, has allocated a certain number of mice. So if you have some variants you've identified and you want to do some follow-up work to understand mechanism, for example, it's possible to have mouse models linked to this program. So this partnership, this research partnership, is about 2,500 people that are registered in a set of domains. They're scattered across the world. These domains have to be led by a UK person who has a medical affiliation, but they're open globally to be members. They have to work as consortium. Think of lots of different ENCODE consortiums working together. It's a shared data space, but we expect people to work together. And they're split according to conditions, and then with some cross-cutting activities as well. Similarly, we have this gene consortium, which has been running as a pilot with a small group of companies, experimenting and seeing what the data is like and how valuable it is. Now, so if you're going to have this lockdown data environment, you have to then be responsible for providing a service to researchers. And that's quite hard. It's a lot easier to make a download link even after you've gone through some ethical approval than it is to provide a service. So this is a hosted environment. It has a virtual desktop interface. So you log in, see a desktop, but you can't see the internet anymore. And you can't cut and paste. Well, you can paste in, but you can't cut out. You can't export any raw data. If you want to export anything out of the system, it has to be summary level data, and it goes through an airlock system, which means people look at those files. You can't export binary because we can't look at it, so you won't be allowed to take it out. But summary level data can be exported for future research and publications, etc. So inside this environment, there are tools. There is an HPC running LSF. And particularly in terms of being able to do some rapid analysis, there are two systems. There's a clinical system called LabKey, which holds all the clinical data and allows you to do basic analytics on that. And then probably more interesting is something called OpenCGA, which has been jointly developed partly with the EBI. This is one of these systems which sucks in all the data from the VCF files into a framework. It's actually got a Hadoop backend as one of the options, and allows you to do rapid queries, so you can specify a number of individuals and then get stats back around the distribution of variance for those subgroups in a few seconds, compared with scanning 100,000 VCF files. So these are the kind of things we expect these GZIPs to do. For one, it's in the title, Genomics England Clinical Interpretation Partnership. There are these, more than 50% of these patients don't have a diagnosis. We'd like these people to help see if they can find a diagnosis. And actually, even now, we've had a few cases where the research arm has made suggestions which have been useful to clinicians. We expect them, of course, to improve algorithms for interpretation. There's all the other things that we might do with this genome. After all, think of this now. These patients have a genome attached to them. That genome's only been analyzed from the point of view of this particular condition, but that genome's not going to change during their lifetime, at least the germline one. What other queries can you make for specific conditions? Obviously, pharmacogenomics is the kind of thing. Decision support linked back into your hospital systems based on alerts around particular variants that may be linked particularly to pharmacogenomic behavior is an obvious place where we could do with better apps that we can deploy inside the system. And then, of course, there's all the sort of standardized research that you might want to do. So this is a generic framework, which we think is probably the future of doing research when it's linked to the medical system. One environment, one data center, running two types of apps: clinical apps for interpretation, feeding back, research apps for researchers. All of the ethics and public engagement and trust is based on the fact that data doesn't move, because people are worried about if the data goes to a company. There have been a number of things in the media recently in the UK, particularly around this, Google and DeepMind having access to patient records and nobody being quite sure what they're doing with them. So this way, of course, it means that we can act on behalf of the health service to ensure that what happens in that data center is all properly ethically controlled and approved, and that we can track what people are actually doing. Now, this does create problems because, of course, you can think now the world is going to move to a whole load of different walled gardens. What do you do in terms of interoperating between them, particular rare conditions? So I think there are several things. Obviously, there's the Global Alliance. That's one option, which is meant to be building APIs to allow federation. So don't share the raw records, share the information with federated analysis. The other option, of course, is to reconsent patients. People with very rare conditions, many times they're happy to be more broad in their sharing. But I think for the health service, if you're going to deploy something across a health service as a whole, you have to start with something which is much more controlled, such as the model we have right now. So there's a few things around, this. I think, one thing about this is how do you get this started politically? I think a key point is that the way we've done this is it's not funded by one side. So Genomics England, for the health service, is paying for some of this infrastructure, but research budgets are also paying for some of this infrastructure. If you try and make the clinical system pay for it all, they just don't want to. They don't want to pay for stuff to support research. And similarly, the research councils do not want to pay for stuff that's involved in clinical treatment pathways. So some sort of hybrid funding model is probably the way to get this sort of thing started in a hospital system. I will skip over this. So I think this is the sort of take-home messages. This structure we've set up with genome medicine centers, it's the engine now for personalized medicine transformation in the UK. By having phenotype data models and making this very standard and a requirement, we are driving secondary data collection in the health system. Because we're using this contracting framework, we really are stimulating activity by small companies providing different types of services. Think of the iPhone App Store. Provide a framework, and then lots of people can potentially, you've lowered the barrier to them coming up with something that can be deployed easily. And all of this is based around this dual data center model, where the data stays in the health system. You can access it, but you can only access it as a reading library, not a lending library. So this is all this is the whole program. 100,000 genomes, whole genome sequences. There's all these activities and industrial partnerships, all part of this effort to drive things forward. And this is funding-wise, is a kind of special project right now, although it is fully integrated. The trick will be to move that from the money that funds these actual genomes to getting that funding as a continuous basis inside the health system, on the basis that collecting a whole genome is cost-effective by health metrics. And that's where we're expecting that transition to happen in 2018. If you want to read more about this, the Chief Medical Officer of the UK, Sally Davis, produced her annual report, which is entirely about genomics. It's quite a big document, but it outlines pretty much everything that we're doing in this whole space. So I'll just acknowledge a lot of people. So, genomics team's about 100. It's actually quite a serious bioinformatics activity led by Augusta Rendon. But it's got big engagement across different aspects of the health system and a lot of support from not just the genome medicine centers, but all the biomedical research centers and a lot of the other cohort activities in the UK. So I will stop there. Thanks very much.
Tim. Thank you for this impressive talk and the impressive effort. It's really... So million questions, but 3 short questions. How much England invested in this, roughly? I know it's...
Okay, so initially it was 100 million to get the thing started, 100 million pounds. By the time you count Illumina's inward investment and all the associated money, there's some money from the MRC, you know, maybe it becomes $500 million, something like that.
Now, second question, do you have plans to sequence normal, healthy people to get a control or to get, background or what is expected?
So we've got biobank for that. So, don't confuse. 100K is the health service. Biobank is a prospective cohort. That's the genotypes of that has just been released. That's 500,000 patients who are, carefully phenotyped. Exome sequencing is now being done with that, and presumably whole genome sequencing will follow. So
for how many people?
Well, I can't remember exactly what fraction. Some of them will get whole genome sequenced anyway. So there are cohorts for background, but in the health service, particularly because we're going to move this to standardized care, we'll only sequence people where it's health justified to sequence them.
And the last question, are people entitled to get their own fully sequenced?
Yes, they can request it. Of course, we will make a significant charge because not only do we have to write it to a disk if you want the whole BAM file, but we have to certify you are who you say you are for ethical reasons. Mm-hmm. It's a whole layer of ethics around all of this.
And people are using it?
I think there's been 1 or 2 cases, that's all.
Thanks for the great talk. I have a question regarding the time point when you do the tests. So far as I understood...
How long?
No, so far, you have to gone through all the different assays and so on that exist in the clinics before you then do this test. So you, for the examples that you gave, for the third example, I think?
Yeah, so there's so in the health system, we've had genetic services since 2003 in a national structure. Yeah. There's about 800 tests which are certified in the health system. The basic eligibility says if there's a standard test, you should have done that first. We don't want to sequence a whole genome if it's in the gene that's known for that condition. But if you don't find it there, we don't want you going on an odyssey. We want you to jump to the 100K.
But then in those cases where, as you said, if we would have known this disease earlier, then we could have intervened earlier. So
that's true. There's a sort of,
if you do it at the last.
I think this is a work in progress. Ultimately, you can see this moving to whole genome sequencing, but in terms of cost and everything, we're not there yet. There is a transformation plan aligned to this idea of switching this, from 100K, which has got a block grant, to being in the health system directly funded, happening in the next year or so. And that will involve a mixture of these sort of things.
So thank you for your talk. How much of control do you have over what's going on to Illumina's facilities? So since they are even doing the variant calling, and since I heard the talk of the Google guy about deep variant call, so I know maybe Illumina and Google might be...
They don't get any phenotype data. Everything is they don't know who these patients are, and they're heavily under contract.
But if they would like to do, they can't do anything with the data.
No, so there's a separation.
Okay.
The health service, you are data controllers. In terms of UK law, data protection regulation, you can contract people to be data processors. That's allowed, and Google was allowed to do that bit. But that's you process a patient and you return the data. That's the limit to what you're allowed to do, and that's a perfectly accepted process, and organizations do it. So that's the framework they're operating under.
So from legal issues, it's clear, but you don't know actually what can actually happen. So
we're the government. Yeah. We know where you live.
Okay, thanks.
Hello, thanks for the talk. I'm Alessandro from DNA Nexus UK. We are a cloud company, and we're routinely analyzing hundreds of thousands of clinical samples in the cloud. And even if this kind of analysis is routine, we always have the same problem at the end. How do you deconsent patients 10 years later when you have amassed information for thousands of them? What is Genomics England's strategy for deconsenting?
Yes, so patients can deconsent, it's true, and essentially because we're not distributing the data, we can delete the data from our system. Now, aggregate data, you can't unaggregate data, but then that's aggregate data, so it's not directly identifiable. So that's the kind of principle, and yes, we have a process for accepting a deconsent and processing, removing the data in that way.
Thank you.
Thank you very much.